it brings that pressure back down. It keeps that pressure really low. So the tissue doesn't stay swollen and bloated. We drain that extra fluid into the lymph capillaries, and that fluid slowly trickles its way back to the cardiovascular system. So how much is it draining? Well, your lymphatic system is draining, what, about 2 liters per day. But that's over a whole day, so that doesn't seem like a whole lot. But think about it, if you didn't have a functional lymphatic system, after 1 day, right, what is that? 2/5 of your blood supply would be missing. It's a good chunk of it, right? So it's a pretty important job. So it's a slow trickle. It's a little volume through each lymph capillary, but it all adds up. It all contributes to this. And as that fluid goes through, you can sample it and sort of figure out what's going on in there, get a good idea about the health of the body. How does everybody feel about how these lymph capillaries work? Pretty good?
Other tissue cells?
These?
Not those.
These.
Like, yes.
Yeah, so these cells represent
Like-
whatever tissue is being served by all these
Yeah,
vessels.
okay. So those are pretty much stationary cells.
They're not going anywhere.
Yeah.
Anything else? All right, what about flow through the lymphatic vessels? Right? This is a really low pressure system. Right? And so you're not going to get high pressures, you're not going to get high flow rates. This fluid's sort of trickling back. But we still need pressure gradients if the fluid's going to move. So the lymph vessels themselves have smooth muscle in the walls, and they can create waves of contractions. Right? That can help propel, almost think of it as sort of massage this fluid back up towards or down to or over to your heart. We also have valves in these vessels. So as you go through the lymphatic vessels, there are passive one-way valves, just like we see in our veins. They work the same way. Keep that fluid going in the correct direction. Don't let it flow backwards. And then we also get to take advantage of the skeletal muscle pump. So not only do we have veins running between muscles, you also get lymphatic vessels. So they get to take advantage of those pumps as well. Okay. I was going to tell you about elephantiasis, but I feel like we should keep moving. You guys know what elephantiasis is due to? Mm-hmm. What's it due to?
Nematode that blocks lymph vessels.
That's right. Small little roundworms that get stuck in there, and then it blocks it, so then you build up fluid in those tissues, and those tissues swell. If you want to talk about nematodes and other cool things, ask me after. All right, let's talk about blood in 2.5 minutes. So, what makes up blood? The components of blood. More than half of your blood, a little more than half, would be plasma, which is mostly water. But in there we also find proteins. We talked about the proteins up here, contributing to osmosis. And there are lots of different dissolved molecules in there. Right? Sugars, amino acids, all kinds of things in that blood plasma. Our hormones are in there. Right? We talked about that with the endocrine system. Those are in the blood plasma. The next most common component to our blood would be red blood cells. A little less than half by volume of our blood are red blood cells. Right? These tiny little anucleated cells. Right? So almost all mammals, their red blood cells don't have a nucleus. If you want to know the exception, ask me after. These cells carry oxygen and carbon dioxide. Right? So these cells are really important for carrying these gases, these respiratory gases. And then we also have white blood cells. This is a very small amount by volume of our blood. But they are involved with body defenses. So they're going to help identify things that don't belong and help attack and break down things that don't belong. And then we also have one more component, the platelets. So these are not cells, they're little tiny chunks of cells. They're a cell fragment. Okay? Everybody got that down? Alright, so this figure here, just quickly memorize this. This shows you the top, a little more than half your blood is plasma, and it shows you all the different things that are in your plasma, lots of different chemicals in there. The cellular elements, not all of them are true cells, but we've got red blood cells, white blood cells, and then platelets, which are little chunks of cells. The white blood cells are actually 5 main types, and even within here there's more variation. I'm not going to ask you to learn or memorize all those. Just know we've got red, white, and platelets. Okay. For that. Let's just show you those proportions. If you took a blood sample, stuck it in a glass tube, spun it in a centrifuge to separate out everything by their density. The plasma, you can see, is a little more than half the volume of your blood. The erythrocytes, those are the red blood cells, a little bit less than half the volume. And this little buffy coat in the middle, that white strip, those are the white blood cells and the platelets. So very little of your blood is actually made up of white blood cells and platelets. All right, the very last thing, I know it's 6:25, but let me just go through this quick so I feel like I finished something. Hemostasis. What happens if a blood vessel breaks? You want to make sure that blood does not continue to leak out of those vessels. So you've got a mechanism that helps stop the bleeding so you can repair that blood vessel wall. 3 main steps to this. Involves vasoconstriction. So your blood vessels are just going to constrict down, which increases the resistance to flow. So less blood flows through that broken vessel. You also form a platelet plug. So those little platelets that are all minding their own business in an intact vessel, once a vessel wall breaks, they all stick together in that broken hole. Let's see how this works. So how do you form a platelet plug? Well, first you get platelet adhesion. In the wall of a blood vessel, you've got collagen. The collagen is not exposed to the inside of the vessel when the vessel's intact. Right? It's sort of hidden. But once that vessel wall breaks, platelets can come in contact with that collagen. And platelets like to stick to collagen. This is sort of what kickstarts the whole thing, what activates these platelets. So platelets stick to collagen. Once they start sticking to collagen, then we go into platelet aggregation. This makes platelets sticky to other platelets. You don't want this to happen in an intact vessel, because otherwise you get these big clumps of platelets and it can clog your vessels. You only want it to happen when a vessel breaks. And when it breaks, it exposes the collagen and it triggers this whole thing off. So the vessel wall breaks, platelets stick in the crack or in the hole, and then more platelets stick to each other in there. So this is an intact vessel. Right? So here's the lumen of that blood vessel. You can see these cells that are shielding the collagen from the platelets inside here. We go ahead and break that vessel, and these platelets right here can now start sticking to collagen, and then they'll start to stick to each other, the platelet aggregation. But that platelet plug is pretty weak all on its own, so we need one more step, and that is coagulation. Coagulation is due to long, sticky strands of protein called fibrin. So fibrin runs through the platelet plugs, and it helps to reinforce that plug. Fibrin is in an inactive form when the blood vessel is intact, and that form is. Install it. Did it roll under again? I put it away? I was ready to get out of here. Fibrinogen. Right? Fibrinogen is the precursor, the inactive form of fibrin. Right? Anything that ends in inogen is a precursor, like carcinogen can lead to cancer. We're going to see some other inogens later on in the semester. So keep that in mind. So fibrinogen gets activated into fibrin, which are these long sticky strands. And then the platelet starts sticking in there, reinforces that plug, makes it nice and strong, and actually contracts and pulls the edges of the blood vessel together. And here's an image actually of red blood cells stuck in the fibrin, right? To those gray lines of the fibrin. They don't care what they stick to. They're just getting everything clumped in there to keep that vessel from bleeding. Hemostasis. That was fast. Questions about that? Yeah, question.
The fibrin contracts and pulls what together?
So once it all forms, it starts to pull together and pulls the edges of that broken vessel together, so it's easier to heal. and it makes for a smaller hole. Anything else? All right, if you've got lab tonight, go empty your bladder and come right over to lab, and we'll talk about what we're going to do next. Otherwise, I'll see you Wednesday. Hi.
A little bit.
Okay, can we talk about this?
Right here. Oh, there you go. Your foundation came off. It's always the same line.
I mean, I have done my part of
This part, I like it except it's a little thick. Or maybe it's just my pores are really crazy. Like my face is crazy right now. Yeah. Why don't you? I use the blender to blend it, but it is. I just use a little bit, and then my whole face is gone.
I haven't done my part where you
So you can go in that lab and just kind of